Sathyabama Institute of Science and Technology.
Good morning, everybody. This is Jesia Preethi, Assistant Professor from the Department of Biotechnology, Sathyabama Institute of Science and Technology, and today we'll be discussing about DNA polymerases. DNA polymerases are enzymes which play a very predominant role in replication, in proofreading the DNA molecules, as well as to maintain the fidelity of the DNA. DNA polymerases have a very crucial and important role to be played. So first and foremost, a DNA polymerase, the main function is to synthesize the DNA, and this DNA polymerases has the capacity of reading the DNA strands from 3' to 5' end. That means that the newly synthesized strand is from 5' to 3' end. Means it keeps adding the nucleotides on the 3' terminal by hydrolyzing, by removing the hydroxyl bond and creating a phosphodiester bond. So DNA polymerase plays a predominant role in making sure that the DNA nucleotides are added and according to the template strand. So there are different DNA polymerases which are present, and prokaryotic polymerases, broadly they're categorized into 5 different types, out of which the first 3 play a very predominant role, and DNA polymerase 3 has a very predominant role in the process of replication. The other 2 have an important role in repair and removal of the primers and resealing the gaps. This is with respect to prokaryotic DNAs. While coming to the eukaryotic DNAs, so there are almost 15 different types of DNA polymerases which have been identified, and out of which we know the role of approximately around 5 different types of DNA polymerases. So first and foremost, if you have to speak about the DNA polymerase which is present in the eukaryotes, DNA polymerase DO is a very important enzyme for the replication of DNA. And apart from DNA polymerase DO, it has, apart from replication, DNA polymerase DO has an important role in repair and proofreading of the DNA which is being synthesized. And coming to the other different types of classic picture which depicts the DNA polymerase, and here this DNA polymerase is something like a clamp-like enzyme which is attached to the nuclear membrane, and you can see there are 3 different domains. On one particular domain, that is the core domain, the leading strand is passed through, whereas on the other domain the lagging strand is synthesized. So DNA polymerase ensures the synthesis of either strands simultaneously. And coming to the different types of DNA polymerases and what are their roles and functions. First and foremost, DNA polymerase DO, as I was just speaking, DNA polymerase DO has a predominant role in replication, and it also, apart from replication, it also has exonuclease activity, which is very important for proofreading. And DNA polymerase alpha is the main enzyme for the synthesis of primers, and also it forms primers for the Okazaki fragments on the lagging strand. And DNA polymerase epsilon is for DNA repair, and it removes the primers which are created on the lagging strands, that is Okazaki fragments, and seals them back with the help of DNA ligase. And DNA polymerase gamma is a predominantly important enzyme for mitochondrial DNA replication. So these are the different types of DNA polymerases which we can find in both pro and eukaryotic organisms. Thank you.